Good afternoon, welcome back. This is the second session of the day, Hot Topics, Best Data from Design to Publish. Our first speaker of this session is Dr. Nathalie Percie de Sert. Dr. Percie de Sert earned her PhD from St. George's University of London and then completed her postdoc work in the field of nausea and emesis at the University of California, San Francisco, and at the Chinese University of Hong Kong. She has a strong research background, including in vivo research, systematic reviews, and meta-analyses. She is currently head of experimental design and reporting at the National Center for Replacement, Refinement, and Reduction of Animals in Research, the NC3Rs. There, she led the development of the Experimental Design Assistant and the AWIC guidelines, the topics of her discussion today. We'll have a Q&A session directly after her talk, so please type your questions into the Q&A box. And with that, I'll hand the presentation over to her.
Thank you very much, Kate, for that introduction. So I'm going to start sharing my screen. Yeah, is that showing my slides?
Yes. Remember, there's a little bit of delay.
Sorry, say that again?
There's a little bit of a delay.
Right, okay, that's fine. Okay, so, well, thank you very much for having me here today. I'm really glad to be part of this 3-hour symposium. And in my presentation, what I'm going to do is I will talk about resources that we've developed at the NC3Rs to support transparent, reliable, and reproducible animal research. And I will talk about the newly revised AWIC guidelines and the experimental design assistant, and especially kind of why we've developed them and the importance of this tool. And then I will do a live demonstration of the EDA. Okay, so I mean, there's quite a few people attending today, and I'm not sure how many of you know about the NC3R, so I'm just going to go ahead and introduce the organization briefly so that you understand where we're coming from. So the NC3Rs is an independent scientific organization which is funded by the UK government. We were established 15 years ago, and we lead on the discovery and application of new technologies and approaches to apply the 3Rs. And partnership is key to everything that we do. So we work with scientists and organizations across the balanced sectors, and that includes universities, other funders, the pharmaceutical, chemical, and consumer product industries, and regulators. And most of our budget goes into funding research, with the main funder of 3Rs research in the UK. But we also have in-house programs that are led by the scientists in the office to embed the 3Rs in policy, practice, and regulations. So we've got programs of work on experimental design and reporting. And in these programs, we basically develop resources to support scientists with the design and reporting of animal experiments. All these resources are available freely from the NC3R's website, and the URL is here on the slides. And experimental design and reporting is really highly relevant to the 3Rs, because if studies are not designed to yield robust results because, for example, they're underpowered because the risk of bias have not been addressed properly, or if studies are not reported in enough details, then they can't actually add to the evidence base. So such studies are a waste of animals, and not to mention the implications when entire programs of clinical work are based on the findings of unreliable animal research. So today, specifically, I'm going to talk about two resources, AWIC guidelines and EDA. And these resources really go hand in hand, and they both focus on the rigor and the reliability of animal experiments. So EDA is an online tool that helps you design rigorous in vivo experiments, and the AWIC guidelines prompt you to report in publication the information that's needed to assess how reliable the results are. So I'm going to start with the AWIC guidelines. And I'm sure a lot of you will actually know about the guidelines already. They were developed 10 years ago to improve the reporting of information in animal research publications. They've just undergone a revision, and the updated guidelines, AWIC 2.0, will be released in the next few weeks. We're actually just discussing the publication date, and that's going to be sometime in July. And the guidelines consist in a checklist of items that have to be included in a manuscript to ensure a comprehensive and transparent reporting. So all these items are important for different reasons. Some of the items are important for internal validity, for example, a description of the study design, how the animals were allocated to groups, how the sample size was chosen. All of this is crucial information to understand how reliable and robust the findings are likely to be. Some of the items are important to ensure that papers contain enough information to repeat the studies that are described. So for example, a description of the experimental procedures and the animal characteristics. And other items provide information about the context and the scientific relevance of the studies. So things like the scientific background, the relevance to the species, or the ethical approval. And these guidelines really apply to all in vivo research, whether using a mammalian species or an invertebrate. It doesn't matter if the species used does not actually require ethical approval, because the guidelines are about rigorous science, so that applies to any experiment. And the guidelines are endorsed by many of the major stakeholders in scientific research. So over 1,000 journals recommend the guidelines in instruction to others. In the UK, all the research councils and the major biomedical research funders were early adopters. So they made reporting publications in accordance with the RAF guidelines a condition of funding, and they were also instrumental in getting the universities on board. And today, many funders recommend the guidelines on both sides of the Atlantic, like the Wellcome Trust in the UK, or the NIH actually mentioned the guidelines in their reporting standards. And we also see significant level of support from universities. So for example, in the US, Stanford or Johns Hopkins recommend the use of the guidelines. And in the UK, most universities have released a public statement of support as well and incorporated the guidelines into training courses, for example. And there's also a lot of grassroots support from the researchers themselves. So over the past 3 years, we've actually distributed 40,000 of those little checklists, printed version of the guidelines so we've had 40,000 requests from 50 different countries. So as I said, the revised guidelines will be released in a few weeks, but an interim version was published as a preprint on bioRxiv last year. And the main departure from the original guidelines is that, first of all, we've prioritized the items into 2 sets, and that's to allow a step-by-step approach in the implementation. So the 2 sets are the ARRIVE Essential 10, and that's the minimum requirements. That's really the basic minimum to include in a manuscript, because without this information, reviewers and readers cannot confidently assess the reliability of the findings that are presented. And we know that journals do not have the capacity to check compliance with the entire ARRIVE guidelines. So here we actually propose an expert consensus on the information to prioritize. And I think this promotes a harmonized approach across journals as well to ensure that all manuscripts contain essential information that's needed to progress the research. And then the second set, the recommended sets, which is very important as well, but there's no point reporting this information if items in the Essential 10 have not been reported. So the Essential 10 is really the information to start with. And we've also updated the guidelines, and we've added a few new items, such as protocol registration here or data access. So these were things that were not really on the radar 10 years ago, at least not applied to animal research. And their importance and prominence is definitely developed over the years, so we thought that it was fit to add them to the guidelines at this stage. And another thing that we've done as well, which is really important, is develop an explanation and elaboration document. So this was published as a preprint as well, and it will be published alongside the guideline next month. And that document is really to explain the rationale behind each item of the guidelines. It clarifies key concepts, especially statistical concepts that are often misunderstood, and it provides illustrative examples of this information, how this information can be incorporated in a manuscript. And that document really provides a lot of information and advice on experimental design. So to make the best use of it, it really has to be read before starting the study, so that you can make sure that you're planning the study in a way that you will be happy to report transparently, and also to ensure that you're collecting all the information that you will need to include in the publication. So revising the guidelines has been an extensive and collaborative effort, with input from the scientific community carefully built into the process. And this is to make sure that the guidelines are relevant to all in vivo researchers. So the revision of the guidelines been undertaken by an international working group with expertise from across the life science community, including funders, journal editors, statisticians, methodologists, and researchers from academia and industry. We also used a Delphi exercise with external stakeholders to maximize diversity in fields of research, a field of expertise, and geographical location. So we had experts from 19 countries providing input. And following their publication bioRxiv last year, the revised guidelines and the elaboration document were road tested with researchers preparing manuscripts describing in vivo studies. So that's to ensure that these documents were well understood and useful to the intended users. And we've also considered other reporting standards as part of the revision to ensure that we didn't provide conflicting advice, which would be confusing to researchers. So ARRIVE 2.0 are compatible with other guidelines that have been published in recent years. And that's because they provide a comprehensive set of recommendations that are specifically tailored to the description of in vivo research. Using the ARRIVE guidelines to write a manuscript actually help authors comply with more general requirements as well. So to explain why we developed resources like the ARRIVE guidelines and why they're needed, I'll use the example of the reproducibility project in cancer biology that shows that for research to be reproducible, it really needs to be reported fully. And this project was set up in 2013, following the publication of reports from the pharmaceutical industry that showed that 80 to 90% of published research could not be reproduced. So the project has already published replication results for 16 of the replication studies in the journal eLife. And the bottom line is next. Most studies reproduced part of the original papers, but also reported some results that were not consistent with the original papers. But what I want to draw your attention on is the number of papers to be replicated. So the study initially set out to replicate 50 high-profile experiments. And that target was reduced to 37 papers in 2015, then to 29 papers in 2017, and in July 2018, a decision was taken to reduce it to just 18 papers. And that decision to cut off some of the replications was taken because costs and resources was spiraling out of control. And one of the main reasons given was that methodological details were lacking from the original papers. So actually, the teams that were set up to reproduce these papers just could not get enough information from them. So clearly there's a problem with the reporting of preclinical research. And there's not enough methodological details in the publication to enable other researchers to reproduce their experiments. But another thing that's frequently missing is information allowing readers to assess how reliable the results are likely to be. And this is a preprint which was published a few weeks ago. And in there, researchers used the SCIENCE core software to analyze 1.6 million open access papers from PubMed Central. And the graph on the left here specifically looked at papers reporting animal research. And they showed that for papers published in 2018, the percentage of papers mentioning the use of measures to reduce subjective bias is very low. Randomization to allocate to group was mentioned in 37% of the publications, blinding in 12%, power calculation in 7%. And that graph on the right here shows the evolution of the reporting of these measures over the last 25 years. So now that's across all papers, not just animal research. So that also includes clinical research. But what we can see here is that there has been some improvement over the years, especially for the reporting of randomization, which increased from 10% in 2000 to 30% last year. But the reporting prevalence of these measures is still very low. And this highlights a problem with the reliability of animal research and whether we can actually trust the results that are published in the literature. So now I'd like to spend some time explaining why some of the items that we've included in the Essential 10 are so important. And I will start with randomization. So the guidelines recommend reporting whether randomization was used to allocate animals into groups, and if so, the method that was used to randomize. And when you allocate animals to treatment groups, the methods is very important because haphazard is not random. So you actually need to use a validated procedure to randomize animals into groups. So for example, a computer-generated list of numbers or throwing a dice, flipping a coin, something that you can't influence. And the randomization has two main purposes. So first of all, it helps minimize selection bias. So it helps minimize systematic differences in the characteristics of the animals that are allocated to different groups. And if you use haphazard selection, for example, you might end up with all the slowest mice in one group and mice in one group. And that's not on purpose, but that's just because you just pick them that way. And randomization also helps you meet one of the key assumptions of the statistical analysis, namely that different groups should be drawn from the same background population using random sampling. So if you've not randomized animals into groups, then actually you shouldn't run any statistical analysis on data. You can't generate a p-value because that's actually not valid. And this graph demonstrates the difference between haphazard and random. So this was a blog which was published on Cognitive Daily, and that's a very simple internet poll where participants were just asked to pick a number randomly between 1 and 20. So there was 350 responses, and each number has got a 5% chance of being picked if it is random, because there's 5 numbers, so 1 in 20 chances. And if you look at generating random numbers using a computer, that's the white bars on this graph. You can see that those actually oscillate around the 5% line, and that's about right. And then with chance, that's slightly above, slightly under, but that's around the 5% line. That's actually random. When you ask people, this is what people did. So 20% of the people picked 17, and a further 10% picked 7. And clearly that's not random, but that actually demonstrates that human beings can't do random. We're always going to overthink it. There might be a reason for picking 7 and 17. 17 is a prime number that seems random, but clearly this is not statistically random. And that's why we need to use a validated method, because we can't actually, we're not geared to doing random. We're always going to find connections. We're always going to overthink it, and we just can't do random. And we can see the impact of randomization in the published literature. So these data are from Malcolm MacLeod's team in Edinburgh. And these are the findings from a systematic review looking at the efficacy of interventions in vivo models of multiple sclerosis. So in this study, they combined the results from 126 different publications, testing the efficacy of 36 different drugs. And they compared the studies which reported that the animals had been randomly allocated to treatment groups to studies which did not mention anything about randomization. And this is what they found. In the studies that were not randomized, the effect size was 40%. So that means that the treatment improved the symptoms by 40%. But in the studies that had been randomized, the effect size was only 20%. So clearly, it was completely overestimated in the non-randomized studies, and the effect size is much more likely to be around 20% in this. So that's what happens when you don't randomize. You actually overestimate the treatment efficacy. Another item in the Essential 10 is about blinding, and we recommend that publications report who was aware of the group allocation at different stages of the experiments. And that's because blinding can really impact the experimental results. And this is a very interesting experiment which was carried out years ago by Wedenzel and Vogt in the 60s actually. And this experiment involved 12 students and 2 different populations of rats. There was a population of maize bright rats that had been bred over many generations to perform well in maize assays. And there was a population of maize dull rat which had been bred over many generations to perform poorly in this type of assays. And then the assay was a simple elevated T-mass with 2 arms. There was a white arm and dark arm. The dark arm was reinforced. So during the training phase, rats were actually trained to go to the dark arm, and they'd get a food reward when they got to the dark arm. And then the assay was performed over 5 days. And the students were recording the number of correct responses, the number of correct turns to the dark arm. So as you can see on this graph, the bright rats started off better than the dull rats, and they improved further during the course of the experiment. And that's all well and good. It was all statistically significant. The issue is that this experiment was not done on the rats. It was done on the students. And the rats had been labeled bright and dull randomly in the morning of the experiment. So the only difference here in this graph is actually due to differences in the mind and the expectation of the investigators. The students actually reported that they liked working with the bright rat better. The bright rats were tamer, they were cleaner, they were nicer to work with. One student even reported getting bitten by a dull rat. And that's what explained that difference here. It's got nothing to do with 2 different populations of rats, but it's got everything to do with different behaviors towards these rats during the course of the experiment. And that's why blinding is so important, because we're always going to project our expectations, and that's going to show in her behavior, and that's going to influence the animal's behavior. And we're just not going to measure what the experiment was set to measure. And blinding is really important for animal experiments because of the behavior towards the animals, but it's also very important for in vitro research. And this is a study which was published in Nature in 1988, which showed that very high dilution of antiserum triggered the degranulation of human basophils. And we're talking about dilutions that were so high that no molecule could possibly be left in the solution. So this is the water memory theory. This is what this is based on, and that supports homeopathy. So Nature actually agreed to publish this paper on the condition that they would send a team to the lab in Paris to observe the experiment being repeated and to see whether the results could be reproduced. So they sent the team to Paris, and haha, surprise, they found that when the experiments were repeated with a strict blinding procedure, so that the tubes containing the dilutions were coded, the slides where the cells were counted were also coded, and the basophils counts were measured in duplicates by 2 independent investigators, there was actually no effect of homeopathic dilution. And I think what's really important here is that we're not talking about a case of fraud. The investigators in Paris were really, really happy to receive that team from Nature, and they really believed that they were onto something. And in a sense, that's the problem. They actually believed it, so they believed it so hard that they ended up fooling themselves. And that's why we need to blind and randomize and minimize any opportunity that we have as researcher to impact on results on purpose or completely unconsciously, because which is the thing, we're not actually doing it on purpose, and we don't realize when we're doing it. And we're seeing the impact of blinding in the published literature as well. So this is the same Systema review that I showed earlier on the effects of drugs in animal model multiple sclerosis. And here they looked at the efficacy of treatment in studies which mentioned that the investigator was not aware of the treatment allocation when assessing the results, to studies which did not mention anything about blinding. And again, they found that the studies that were not blinded completely overestimated the treatment efficacy. The inclusion and exclusion criteria are also crucial information to include in the paper, and they're included in the AWIC Essential 10. And Scott and colleagues provide a very good case for it in the analysis of a common model of ALS in mice. So this is a model that has shown very poor clinical translation. Many treatments that have been shown to work in mice do not show any efficacy in humans. So the researchers analyzed the historical database of animals that had been used as controls in efficacy studies to investigate the reasons for this discrepancy. And the data set was quite noisy. There's lots of variation between animals. And they used computer modeling to explore this variation and identify what variables were responsible for the variation. So they sampled animals at random and they assigned them to groups. And by chance, they obtained differences in survival time between the groups, even though none of the animals had received any treatment. And this graph here shows the frequency at which group differences were seen. And you can see that it depends on the sample size. And the smaller the sample size, the higher the chances of seeing a difference between the group. And when the simulation was run with 4 animal per groups, effects were detected in 55% of the time. And with 20 animal per groups, the effect was seen in 25% of the time. But when appropriate exclusion criteria were applied, this frequency reduced drastically. And when they excluded animals that died from non-ALS causes and animals that did not contain enough copies of this transgene, then at n equal 4, exclusion criteria reduced the frequency of apparent effect from 55 to 35%, and at n equal 20, it was reduced to less than 5%. So it's likely that all these published studies that had reported that the treatment worked actually had detected noise rather than actual effects of the drug. And that's because they didn't use appropriate exclusion criteria. So the new AWIC guidelines will come with a new website. And that's the link to the new website here. Right now, this link takes you to the AWIC page on the Institute's website, but we are preparing a brand new website that is completely dedicated to the guidelines, which launch when the guidelines are published next month. And as with the current website, this will include many freely available resources. So we'll have fillable AWIC checklist. So these are used to indicate in which section of the manuscript the relevant information can be found. And some journals request this checklist as supplementary information. So we'll make these available for the Essential 10 set only and for the full AWIC so that the journal can actually tailor the requirements. We'll also have translations. The original guidelines are available in 8 languages. We're currently working on translation of AWIC 2.0. The website will also include explanation and example for each of the items. And in the next few months, we'll also add new resources. So we're currently working on an operationalized checklist to assess a manuscript compliance with the Essential 10. And that's something that could be very useful to editors and journal staff. So these are resources that we develop in the next few months and year, and that we'll add to the website gradually. So now moving on to the Experimental Design Assistant, or EDA. So the EDA is an online system to help researchers design in vivo experiments. And I just want to reiterate that the EDA really goes hand in hand with the RAB guidelines, because when you're designing your experiment in the EDA, it actually helps you capture most of the information that the RAB guidelines recommend reporting. This system is freely available, it's online. You can access it on eda.ntqrs.org.uk. And the EDA is really like your own personal statistical assistant. So it provides bespoke advice and feedback on your experimental plans. It gives you recommendations on statistical tests that are appropriate for the design of your experiments. And it provides dedicated support for randomization, blinding, and power calculations. And just very briefly to introduce the system before I go into the demonstration. So this, in the EDA, each experimental plan is represented as a diagram. And that might take a little of getting used to. So I'm just going to talk you through that diagram. This is a very, very simple two-group comparison. But basically in this experiment, a group of mice is split in two. Group 1 gets a vehicle injection, group 2 gets a drug injection. Then a measurement is taken. In this case, plasma glucose levels are measured and the data is analyzed. The independent variable of interest is the drug with 2 levels, vehicle and drug. So that's very, very simple experiments. Inside each of these colorful blobs, you get more information. So that makes the diagrams a lot more explicit than text descriptions. If you think of blinding, for example, in a text description, best case scenario, you're just going to have experiments was done blind, and you've got no idea what was blinded, who was blinded to what actually happened during the experiment. On an EDA diagram, the blinding status will be indicated in the allocation node, in the measurement node, in the analysis node. So you know exactly what step of the experiment are done blind. So it's perfectly acceptable not to do everything. There's always going to be constraints in animal research. If you're working with white and black mice, or very obvious differences in phenotype, it's going to be very obvious which ones are which. So you can't actually blind the measurements, but there's no reason that you can't blind the analysis. So it's about displaying that granularity and being transparent about how the experiment is going to be done really in practice. And the diagrams include 3 parts. So you've got the high-level information about the experiment in gray nodes. In blue and purple, you've got the practical steps. So that's really what's done in the lab. So you've got groups of animals and you're splitting them. You give them treatment, interventions, measurements. So it's all about the practical stuff. And then the green and the red parts are the analysis plan and the variables in the analysis. So the entire experimental plan is actually included in the diagram. And you can represent any type of experiment as a diagram. So we've developed an ontology which works a bit like Lego bricks. So, we've developed the bricks and you can combine them in any way that you want and you can represent anything that you want with it. So it's the same principle. And then the system uses computer-based logical reasoning to provide feedback. So for example, that would be an example of a feedback prompt asking you to clarify the blinding status when the results are assessed. So in this prompt, you get an explanation as to why blinding is important, the different stages of the experiment that can be blinded, and the different ways to blind different stages of the experiment. So it's just giving you enough information so that you can make an informed decision on what's appropriate for your experiment. So the EDA has got loads of benefits, and the main goal that we had when we developed the system was to improve the reliability of published results. And the EDA does that by addressing obvious sources of bias and providing advice on how to address them and account for this bias in the analysis. It also promotes a better understanding of experimental design, and it raises awareness about why these issues are important. And every time a user gets feedback from the system, they actually learn something new. So the system is certainly not a black box telling you what to do, but it's actually providing you just enough information about the implications of making design decisions on your experiment so that you can actually understand why you're designing your experiment a certain way. And it's just giving you enough information. It's not going to, send you to read an experiment design book. We've actually just, broken this information in bite size so that you can get just enough so that you can make the decision and you can decide what's the best thing to do for your experiment. The system also facilitates peer review and assessment of the experimental plans with an explicit description. So I said that those diagrams are a lot more explicit than text description, and that's very useful at the level of a grant application, ethical review, manuscript submission, or even a published paper. That transparency, this granularity is really useful for editors or peer reviewers or readers to actually assess what, how it's been done or how it's going to be done. It's also useful as a form of pre-registration. So the diagrams contain all the relevant information that you'd want to pre-register. And you can actually provide this as evidence that a study was planned as you're reporting it, and that, for example, you've not changed your primary outcome measure based on the results. And the diagrams really, and the system on its whole, really promotes more careful consideration of the experimental plan. So it really forces the scientists to think about the experimental plan, identify the variables, consider the data that's going to be collected. And we want scientists to spend a lot more time planning their experiments and think about them carefully. And it's much better to do that than thinking about analyzing the data once the data has been collected already and there's nothing that you can change. And a frequent question that I get about the EDA is, how long does it take to plan an experiment in the system? And to be honest, if you've actually already identified the plan, if you've got everything, if you've got all your variables, you've got everything planned out, it takes 10 minutes to put it to the system. Putting it to the system is not what takes time. What takes time is actually thinking about the design, is getting feedback from the system, thinking about that feedback, maybe changing the plans, discussing the plans with your collaborators or your colleagues. That's going to take time. That's fine. That's actually time well spent. And I think we should be spending a lot more time planning experiments, and that would actually remove a lot of headache down the line. And we know that the diagrams are also used to discuss the plans in lab meetings. And I think having that visual representation really facilitates discussions. And normally in the lab meeting, you're just going to show your results, right? You just put your graphs up because you've got something visual to show. But by the time you've got your results, you can't change anything. So feedback from your peers is really not going to make any difference. Whether with the EDA diagrams, we know that people are actually using them in lab meeting. And that's really, really useful because you've got a layout of the experiment straight away. You can see in one glance how many groups you have, what you're doing with the data, how many transformations you're doing to the data, and so on. So you can actually ask the hard questions. Whether if you're just listening to someone describing an experiment, 10 minutes later, you're still trying to remember how many groups they said they had, and you're just not going to be focusing on the important questions here. So the diagrams actually facilitate that discussion with the visual representation, and everyone's clear as to how the experiment is going to be done. So the PI doesn't find out 2 years down the line that the experiment was not actually run the way that they thought it was going to be run. And just a quick one on the uptake of the system. So this is a system that was launched in 2015. And there are now 9,000 accounts on the system. The website is visited about 3,000 times monthly. It is recommended by 80 organizations in 70 different countries, mostly funders, because funders actually see the benefit of the system and they can see the value of funding, good, well-designed experiments. 25% of the users are actually in the US and a further 20% in the UK. So that's what it looks like in terms of usage. If you're interested in the system and you want to read more about it, I recommend that publication in PLOS Biology, which provides a lot more detail. So that's the end of my presentation. So I'm going to move on to the live demonstration. So I'm going to have to stop sharing my screen for a minute and change what I'm showing. Hold on. Okay, so I need to share Chrome. Okay. Right, so you should be seeing my screen here. So this is the homepage when you get to the EDA website. So the EDA is a software which is hosted online, so it's actually hosted on a website. So that's the website when you get to it. And on this page you get, just a brief description of what the system does. There's also a link to more information on how to use the EDA, for example, in support of funding applications or ethical review. So there's a lot of information on the website itself, even if you're not going to be using the EDA app yourself. Another part of the website which is really useful, again, which is just on the website if you're not using the app, is about experimental design. So actually, it's a very comprehensive resource on the different concepts that are used in experimental design. So we've got loads of information on variables, on group size, experimental units, allocation, blinding, and so on. So there's a lot of useful information or useful contents that we've developed that you can refer to. But then if you want to use the EDA itself, then you go to the start page. If you've got an account, you can log in or you can apply for an account. I just wanted to highlight that we really need minimal information to register for an account. All we need is a username, an email, and a password. It doesn't have to be real name. It doesn't have to be your institution email. If you want an anonymous account, you can do that. That's not a problem at all. So I'm just going to log in. And so that's the landing page in the EDA. So from there, if you've never used the system before, you've got all the right links and all the right information that you need. So there's links to how to get most of out of the EDA, the kind of like the sequences that you'd go through. So you start by drawing a diagram, adding details into the properties, critiquing your diagram, getting feedback. Then you can choose a method of analysis, calculate your sample size. So you got all these steps that you can go through. So you can read that if you've never used the system before. That will give you a good starting point. We've got video demonstrations, we've got a user guide as well. If you've not used it before, I recommend using templates to start with. So we've got a battery of templates and examples, and those are really useful because you can just load a template and then customize it to your needs. So the templates are going to be completely empty of information. And each of the templates is linked to an example just to put this information in context. But if you choose a template, you have to choose a template based on the design of the experiment. So I wouldn't want you to choose this one because you're doing glucose studies, but it be chosen because you're doing a two-group comparison. And we've got several templates to choose from. So a two-group comparison, this would be a crossover design with washout periods. This one would be a comparison between genotypes when you're not actually actively randomizing animals into groups. This one would be an experiment with several different factors. So you're looking at the effect of exercise and sex, for example. And this one would be a three-group comparison. So you've got loads of templates that you could choose from. As I said, for the purpose of this demonstration, we're actually going to start from scratch so that you can get an idea of how that works. So I'm going to go back to the start page. And I'm just going to start from scratch. Yes. So that's where you build your diagram. That's the canvas. And you can build your diagram using all the nodes that are in this column here. So as I said, you've got different type of nodes. So you've got the high level information, the practical step, and the analysis nodes. If you're not sure what we mean, we've got all these names here. If you're really not sure what the outcome measure here, you've got little question marks here that you can click to get help. And if you click on the outcome measure, it will actually give you more information as to what is also known as. It's also the dependent variable, the response variable. It will actually explain what this is, just to help you recognize what it is that you're working with in your experiments. So all the nodes are actually explained in the system as well, in the Help Center. And then it's going to explain all the information that's captured as part of the outcome measure. So you've got explanation here as well. So I'm just going to go ahead and just reproduce that simple two-group comparison that I showed earlier. So I'm going to start with the high-level information. So I'm just literally just dragging and dropping onto the diagram. If you open the node, if you look at the properties here, that's the type of information that you'll have to provide here. All the information that's got a little black star is information that's crucial to the internal validity of the experiment. So you have to provide it. If you don't know, don't try to guess. Just leave it blank, and then actually the system will help you provide this information. So don't try to guess anything. If you're not sure, just leave blank and wait for the feedback. Kind of information that you provide here, the hypothesis, the effect of interest, the effect size, justification for effect size. So that would be the high-level information. You could add animal characteristics as well. So in there, you'd specify the species, the strain, the sex, the weight, and so on. If you're working with two different, for example, male and female animals, then you'd add two different animal characteristics nodes so that you can provide the detail for each type of animal that you're including in this experiment. And then let's go to the practical steps. So again, I'm just dragging and dropping. So we're starting with the group. So this one would be the pool group. I'm just going to double click on it to edit the label. So that's my pool group. If you want to edit the label, you can also do it from the properties here. So this is the type of information that you have to provide for a group. I'm just going to leave it blank for now. I'm just going to build the diagram and we'll look at that later. And if you look at that menu here, that's the system will actually help you build that diagram. So it's actually giving you the step that you can add after that. So we want to split this group. We actually want an allocation. So if you hover over these icons, it will actually tell you what they are. So this was an allocation. So this is exactly what we want. And we want to allocate this into 2 groups. So I'm just going to click on the group icon. I want 1 group, go back, and another group. So that's our 2 groups. And then each group was subjected to a different intervention. So group 1 was going to get the vehicle. So that would be a pharmacological intervention here. And group 2 was going to get the drug. So that's a different pharmacological intervention. Then the animals were going to be measured. So I'm looking for a measurement. So here it is, measurement. And they're all actually measured in the same way. They're getting different interventions, but they're all measured in the same way. We're going to measure plasma glucose levels in the same way in both groups. So instead of doing a different node, I'm just going to link this one. And I'm just going to use the little arrow here and just drag it into that node to link it. And as you can see, the system is actually giving you feedback. It's giving you green corner saying, yes, you can do this. You can drop it, and that's going to link properly. If I tried something else, it actually gives me feedback saying that's not right, you can't do this, that does not make any sense. So you get a bit of guidance in that way as well when you build your diagram. And then, so I've got my measurement, that's going to be, so I need an outcome measure here, so looking for the outcome measure, here it is. And then the data will get analyzed. So, I've described it a fair bit. I'm just going to critique and get feedback from the system, and the system will actually help me build the rest. So to get feedback, I'm just going to go to Tools and Critique, and that's going to take a few seconds. If you need more room, you can just collapse this, so you get a full view of your diagram here. You've got, if you look at the menus here, you've got a view menu, you can zoom in and out and so on. So that's going to take a few seconds. Here it is. So that's the feedback. So we've got 6 hours and 15 warnings and no advice at this stage. So the feedback actually comes at the level of different nodes. So all the red ones are errors, the yellow ones are warnings. So I'm just gonna, the warnings are gonna be because there's information missing in each of these nodes. So that's fine. We're just gonna ignore that for the purpose of the demonstration. I'm just gonna deal with the errors. So that's the first error. So we've got 2 errors here. The independent variable of interest is not specified and the experimental unit is missing. So I'm going to look at further detail. So in this prompt, I'm getting information as to what an independent variable of interest is, common examples of independent variable of interest in an animal experiments, for example, a drug, surgery, sex, and then I'm getting advice on how to actually add this to my diagram. So you can read this prompt on your own time. But basically that makes me realize, yes, I'm actually using a drug. That's my independent variable of interest. So I'm just going to follow the advice here. And I'm going to add my independent variable of interest. So going back to here, I'm just going to drag the independent variable of interest onto the canvas. I'm going to edit the label to specify that's my drug. And we've got 2 levels. We've got drug and vehicle. Oops. And drug. Okay, and the advice in the prompt was actually to connect this to the analysis, so just to show that's a factor of interest in my analysis. The second error was the experimental unit missing, so I'm looking at the experimental unit, so that here I've got guidance on what the experimental unit is, and just information that will actually help me recognize what it is. So, it could be the animal, could be the cage, could be the litter. So I can read this and actually realize that actually in this experiment, I'm injecting all my animals independently. The experimental unit is the animals. So I'm just going to follow the advice in the prompt, add an experimental unit node, and specify that my experimental unit is the mouse. And I can use the drop-down menu here to specify this is an animal. So 1 unit is 1 animal. Okay, we've got more errors. So here it's telling me that my groups are not differentiated. So the system is actually not seeing the difference between group 1 and group 2, like what are they actually, what differentiate them. And the way that you indicate this to be explicit on the diagram is by tagging with the tag. So it's very clear which group is receiving which treatment. So you can read the prompt in full, but I'm just going to follow the advice quickly. So I'm just going to select these nodes by dragging over them, and I'm just going to copy and paste. That's the easier way of doing it. And then, so group 2 is going to get the drug, and group 1 is getting the vehicle. So here's my diagram. So there's one more here that I'm going to deal with. So outcome measure, I've not specified whether this was continuous or categorical, and whether, and actually I've not specified what it was at all, so we don't, it's just this outcome measure. I'm actually going to have to say I'm measuring plasma glucose levels, because outcome measure doesn't mean anything. So that's my outcome measure, plasma glucose. And I'm not going to open the prompt, but basically in the prompt you'd get information on how to recognize whether your data is continuous or categorical, and the implications of working with continuous or categorical data. So in this case, that's continuous. I can ignore this one. It's the primary outcome measure. I only have one in the experiment, so it doesn't matter. Okay, so I've dealt with a fair bit. So I'm just going to critique again and see whether the system's got more feedback for me. And you will go through this, a few times, because basically the feedback is going to update every time. So if you've added, or if you have added more information, actually you might get more feedback, because the system is detecting more room for improvements. So now I've got 2 errors in 15 warnings. So actually the 2 errors are on that new nodes that I've just added and the new independent variable of interest. So I need to specify whether it's continuous or categorical, and I need to specify whether it's a repeated factor. So just to look at one, as I said, here you get information helping you to recognize whether you're working with a continuous or categorical variable, the implications of working with continuous or categorical variable, because actually it's about treating it as continuous or categorical. So that's for you to make that decision, really. So you can read that on your own time. For this experiment, I'm gonna treat it as categorical. So here it is, categorical and repeated factor. I've not opened the prompt, but this is not a repeated factor. Do we have anything else? I'm just going to open one of the warning to show you. So we've got one here that's quite interested, saying that this diagram does not include any nuisance variable, and that's actually very unlikely for an animal experiments, because normally there are other sources of variability that needs to be accounted for. So you can read that prompt and give you a lot of examples of common sources of variability that needs to be accounted for in the design of experiments, and how they can be accounted for. So for example, here, this one, it's talking about the day of the experiment. You're like, oh yeah, this experiment is actually going to do over, it's going to be done over different days. So you can read more information that sends you to the website, and you've got loads of information on, nuisance variables and how to account them as blocking factor. So you can read that in your time, but that, that might prompt you to realize, yes, actually, my experiment is going to be done over several days. So I'm going to follow the advice and add this nuisance variable in my experiment. So I'm going to add this nuisance variable, that's the day, and that's going to be done over 2 days. So let's go with day 1 and day 2. Okay, and the advice was to add this into the analysis. Here it is. You can see that the link is red. That's because there's actually more than one ways that you can add this to the analysis. So the advice, so I'm just going to highlight it, and then on the, if you hover the red spanner, it actually gives you a choice. So I'm going to select blocking factor, because that's what the advice was saying earlier. Okay, so I'm just going to critique one more time, see what we've got. And the first round of critique will kind of help you build the diagram. And then as you get more specific, then you get more and more critique about the design itself, and kind of, helping you set your variables. And so we've got one more error still. Let's look at it. There's one more error on the nuisance variable. So that's what I was saying. So it's actually, it's actually identified that I was trying to use this as a blocking factor, but that means that this nuisance variable needs to be categorical. So then I get a lot of information on categorical blocking factors. Is this really what I want to do? And then there's another option, actually. If I wanted to treat this as a continuous variable, this would be the implication. So I can read this, decide, okay, now I'm actually quite happy with this. I'm going to treat that as a categorical blocking factor. So I can just input this information in there. And all of this information is going to be used to provide a recommendation for the type of analysis that I can run on this data. We've got a warning here on this new nuisance variable. So So let's look at this one, for example. So it's telling me that I've included a blocking factor in my analysis, but actually I should be included in my randomization as well. So then I've got, it's giving me all the implications of including it in the analysis, which is actually to address the variability. But then, I'm not actually addressing potential bias by including it in the randomization. So I'm going to follow the advice, include this in the randomization as well. So I'm just going to connect it to the randomization, and here it is. So again, it's red because there are more than one option. So I'm just going to have to select blocking factor. I'm just going to tidy up the diagram. So if I just click on the link, I just bend it. Here it is. Okay, so I think I've addressed all the errors. So now I can show you how to get an analysis recommendation. So you just go to Tools, Analysis Suggestion, and it's just going to analyze the diagram and provide me with a suggestion for an analysis method that is compatible with this design. It just takes a few seconds. There it is. So we've got one suggestion. So suggestion is found on the analysis notes. So if I open it, so it's suggesting that I should analyze this data with the one-way ANOVA with blocking factors. And then in there, I've got information about the parametric assumptions and how to test for them. And then if my data doesn't actually meet the parametric assumptions, then what data transformation I can apply to that data. And if that still doesn't work, then I've got recommendation about rank transformation. So I might still be able to use a one-way ANOVA. And then there's information just to remind me as to what the ANOVA can actually do, what question that's actually answering, to help me decide, to help me certify that's actually what I'm trying to do here, and help me check that this is the right way of designing these experiments and analyzing this data. And I've got recommendation of statistical software that I can use to run a one-way ANOVA with blocking factors. So we've only made like a couple of suggestions. There's always going to be one suggestion, which is a free software, like in VivoStat, which is a free software that is specifically designed to analyze animal experiments. And you can just download it freely online. And then we've included kind of very common softwares as well, but you're welcome to use any software that you're normally using as long as it's providing that kind of test. And then we also have something on this particular prompt. We also have something about a dose response experiment. That's because we know that dose response experiments are often analyzed in this way, which is not appropriate. So you get one more chance to actually read this and realize that if that's what you're doing, actually, there's a better way of designing your experiment. So then you can actually make changes at this stage if we've not caught it before, basically. So just very quickly, because I'm actually running out of time. So now that we know how we analyze the data, we can look at how many animals we need per group. So we can go to Tool, Power Calculation. We only have 2 different tests on the system, but they come with full guidance on how to use them. And when I say full guidance, is actually explanation, power calculation, a decision tree helping you to choose what calculator you should be using. And very often that actually boils down to a t-test, either pair or unpaired. And so you can actually go through this to check what calculators you should be using. And then we've got a lot of information on the parameters in the power calculation. So how to decide on your effect size, if you need to be using CoNSD, how to do that, how to estimate your variability. And we've actually ranked it in the most reliable to the acceptable, basically, because depending on what data you've got access to. So you've got all this information that you can check that will help you just do your power calculation. So, let's say that you've all read it. Now, our effect size, so let's say that a difference of 2 millimole per deciliter would be relevant, biologically relevant for this experiment. So that's going to be a minimal effect size. Let's say that my variability from a previous experiment was 1. I'm going to leave the significance level and the power as it is. And I'm just going to calculate, I need 7 animals per group. So close that, I can just enter that in the on the diagram. So I need 7 animals. That's actually 7 experimental units because animals and experimental units are the same in this experiment. So I'm just going to input that in group 2 as well. You can input the justifications. I could actually type the parameters that I've used from the power calculation here. If you've entered it once, that's enough. You don't need to enter it in every group. You just need one justification, and that's going to be enough. You're not going to be tested about it. Okay, so I'm going to close that. So now that I know how many animals I need, I can look at the randomization. So I'm just going to save that quickly first, and I'm going to go to Tools, Randomized Allocation Report. So as I said, it's helping you with the blinding. So it's not going to send you, it's not going to give you the randomization report. It's going to send it to the person who's going to help you with the blinding. So just to show, I'm going to email it to myself right now. But what you should do if you're actually running this experiment is that you email it to someone who you trust, who's going to help you with the blinding. And all you get is just a confirmation that it's been done. So 16 animals have been randomized into 2 balanced groups, and day 1 and 2 have been randomized separately. And I generated one before, so I can show you what that looks like. Where's my Excel?
And Natalie, just a note that your time is almost up.
Yes, that's the last thing that I'm showing.
Okay.
So that's what a randomization report looks like. So the person who's helping you with the blinding, so the person who's going to be coding your syringes, for example, or injecting the animals, the technician who might be doing that, they will receive this, and they're just going to have to input the unique animal identifier for group 1 and group 2, and they're allocated to 2 groups. So I think that's all I wanted to show. I'm happy to answer any questions. And there are video demonstration of it on the website if I've gone over a few things too quickly. So I'm just going to close that and stop sharing my screen. There we go. And I can answer any questions.
Okay, I think we have time for one question. We have a question about the use of EDA, and the comment is that it works well for basic evaluations of a drug or placebo, but people are having trouble using it with a randomized block design. Can you talk a little bit about that?
Yes, well, so the demonstration that I've just done was a randomized block design. So okay. I think that might answer the question. You can do, you can do block randomization in EDA. That's not a problem at all. And there are templates showing that. So the issue might be because they're not sure on how to represent this. And I would recommend having a look at the templates and the experiments that are in the system already. Otherwise, we're more than happy to look at specific design. So we're happy to look at the design, and if they can contact us, and if you else would be helping.
Wonderful, thank you. Can you talk about how the old, arrive, and new guidelines have changed in the way that studies are reported and how you measure the impact?
So the new guidelines are not even released yet, so we don't have any impact of them, obviously. I think what we've actually done with the revision is enable that metrics to be recorded, because we know that the original arrive guidelines are not as effective as we want them to be. There's only one study that's really looked at it in detail, which is the Icarus randomized control trial, which was carried out a few years ago in +1, showing that when you ask authors to fill in an arrive checklist, and you just don't make any other change to the editorial process, and the editors are not even aware that the study is happening, that actually does not improve reporting. So this was a very specific trial of an intervention. It doesn't say that, you know, if you made another intervention or if you trained reviewers, you might see an effect. But the issue is that this study was a massive study, which was carried out over several years just to answer that one question. And the arrive guidelines, in the way that they were designed, are not amenable to metrics. It's just very difficult to actually look at all items of the arrive guidelines. Some of the items are subjective. So what we've done in the revision is actually isolate this essential 10, which are very, very objective criteria. And we'd be releasing an operational checklist that actually help people assess the impact of the guideline, assess the compliance or the adherence with the guidelines in published papers. So basically with the new guidelines, we're making this possible because it was just not feasible with the guidelines as they were designed before.
Wonderful, thank you.